My talk is going to focus a little bit more on the technology side of genomics and sort of take you through what's been going on in this meteoric rise in information that's being generated in labs around the world regarding genome sequences. So the outline of my talk is I'm going to tell you about DNA sequencing and how it works and how it's really changed very dramatically in the last few years and is continuing to change. I'm going to talk just a tiny bit about a research project that my lab is involved in that actually applies the DNA sequencing technology to studies of transposable elements, or so-called jumping genes, which we think are important in some of the complex traits that Dave alluded to. And I'm going to end up transitioning from the age of reading or DNA sequencing to what I see anyway as the next age of genomics, which is the age of writing DNA sequences and actually synthesizing DNA to order. So looking into the future a bit. Okay, so a brief history of genomics. You all know that in 1953, Watson and Crick first proposed the double helix structure of DNA. Back when I was a graduate student, DNA sequencing first appeared on the scene. 25 years ago, a bacteriophage, small bacteriophage, 5x174, 5,000 base pairs was sequenced. That was a revolution at the time. And as you can see over the years, larger and larger genomes, you can see the orders of magnitude on the genome size creeping up until 2001, human genome draft, 3 billion base pairs. A few years later, the so-called next generation, the first next generation sequencing machine called the 454 sequencer was released. And this is one of those factors of 10 that Dave alluded to. Dave's already talked about the fact that we're way beyond a human genome now. We're in the 1,000 genomes phase, and obviously it's not just 1,000, but many thousands. And about a year ago, a third generation of DNA sequencing machines was released that again promises to push the envelope even further. So just my take on this, and this is a very important aspect of DNA sequencing technology and why we have so much information, is it's all about the cost. So between 1999 and 2009, there was a 19,000-fold drop in the cost per base pair. So I'm just reiterating what Dave has already shown you in the diagram, but this is really these remarkable changes in technology are what have enabled some of the profound increases in information that we have about human and other genomes today that we could only have dreamed of many years ago. Okay, so to explain to you a little bit about how DNA sequencing actually works under the hood, I've given you the easy version of how you make 2 DNA molecules from one molecule, because this is really what's at the core of all of the DNA sequencing technology that's in use today. So over here on the right, you see a parental DNA duplex that's in the process of being replicated. And Watson and Crick actually recognized this when they first saw the structure of the DNA molecule, that intrinsic to that structure, there was a mechanism for replicating the molecule to form 2 daughter molecules. And that's because, as you hopefully remember from Biology 101, the language of DNA has 4 letters, A, G, C, and T. And a simple pair of rules: A always pairs with T, and G always pairs with C. And so the different colors that you see there, red and green, could be A and T, and yellow and blue could be C and G. You can see that as you add on individual building blocks called nucleoside triphosphates, you can see that this red base here is going to pair up with its green counterpart and thereby properly replicate the DNA. And the enzyme that actually carries out this reaction is DNA polymerase. And DNA polymerases, of course, are an essential part of all organisms, essential protein in all organisms that carries out this process. So this, it may look complicated to you, but this is actually the very, very simplified version of what's going on. So you may also have heard the term anti-parallel relating to DNA structure. And what that means is that the two strands in the DNA are chemically sort of pointing in opposite directions, which we call 3' to 5' and 5' to 3'. And that creates sort of a topological problem for this molecule of DNA polymerase as it goes down. There are actually two of them that have to coordinately replicate these two strands. So here comes the complicated version, and this is a animation of what we think the replication process actually looks like. So at the front here, you see a spinning blue disk. That is called a helicase, which unwinds the double helix, producing two daughter strands. And that spinning molecule there is rotating at about 10,000 RPM, which is approximately equivalent to the rotation speed of a turbine fan in a jet engine. So this is an extremely rapid process, extremely dynamic process, and you can see there, the two outriggers here are two different DNA polymerases. One stays fixed and continuously makes new DNA, whereas this other one over here has a much more complicated task where it connects and disconnects as the DNA is spooled through. So anyway, I'd be happy to answer questions about that if you're interested in more detail. Obviously, we've learned an incredible amount about this very complex process. But the heart of all of this is that DNA polymerase that adds one base at a time. So this is actually the original type of DNA sequencing that was done for the first human genome is called Sanger sequencing. And this is an exhibit from the Broad Institute. If you go up to the Broad Institute in Cambridge, Massachusetts, you'll see this in the window. And what you see here is 96 simultaneous sequencing reactions that are coming off of a machine that's up there in the left-hand corner, which is now obsolete. And you can see that the color code of the four nucleotides there is actually flowing out of one machine. This is an enlargement of what's going on over here. And this is approximately 10 times speeded up, but this is the kind of data that would come out of one of these machines at the Broad Institute. This happened to be sequencing the rabbit genome. Okay, so this is the Broad Institute, and you can see they bought a few of these machines. And this was one of the things that helped them be one of the major players in completing the human genome. All of this is obsolete. The Broad is one of the few places that has a real factory floor type approach to doing science. It's very industrial, and that's what you need to crank out a lot of information.